It's my very great pleasure to introduce a colleague and good friend, Marla Sokolowski. If you have read the bio sketch that was in the announcement for today's talk, you will have seen the many distinctions and awards that have descended on her, like manna from heaven. To name just a few: university professor at the University of Toronto, fellow of the Royal Society of Canada, the Flavel Medal for Biological Research from the Royal Society of Canada, only the second woman to receive this award, and many others. Just a few months ago, she received the J.J. Barry Smith Doctoral Supervision Award from the School of Graduate Studies in recognition of her outstanding role in supervising 40 graduate students and 20 postdoctoral fellows. Even more recently, another distinction is that she has been summoned to the aid of the Department of Ecology and Evolutionary Biology by being installed as its acting chair. I hesitate to call this an award, and we do hope that she will emerge unscathed. There has to be a reason for this impressive evidence of recognition. I think the most cogent explanation is that she has made significant discoveries that provide insights into a major enduring question, namely, is behavior determined by inheritance, or is it mainly driven by experience and environment? This is often referred to as the nature or nurture dichotomy. Back in the time of Queen Victoria, there was widespread acceptance of inheritance as the major factor. For example, if you have read the Sherlock Holmes stories, you may recall that in The Adventure of the Copper Beeches, he made inheritance-based deductions about the character of the villain by analyzing the character of a young son as follows, in quote, "My dear Watson, you as a medical man are continually gaining light as to the tendencies of a child by studying the parents. Don't you see that the converse is equally valid? I have frequently gained my first real insight into the character of parents by studying their children. This child's disposition is abnormally cruel, mainly for cruelty's sake. And whether he derives this from his smiling father, as I suspect, or from his mother, it bodes evil for the poor girl who is in their power." Marla's more recent observations during the time of our Queen Elizabeth began in the 1970s when she was a graduate student here at the University of Toronto and discovered a gene in the fruit fly, Drosophila, that regulates feeding behavior. Building on this discovery, she has uncovered significant environmental impacts acting on the genome through what are termed epigenetic processes. So although genes are important and constrain us in many ways, they do not have absolute veto power in determining our natures and behavior. Marla will discuss this, and we look forward to having our mental horizons extended. Without further ado, let the fun begin. Marla.
Sorry to talk my way through this, but it sometimes works better when you do. There we are. And we've got that nuisance at the top, and I think we're going to live with it because I don't know how to see it and get rid of it. Okay.
Thank you for saving the day, Daphne, and Harold for that wonderful talk. And I love Sherlock Holmes stories, so I'm going to pay more attention to that. Okay, well, good morning, everyone. Thank you so much for inviting me. It's a pleasure to talk to this esteemed group. And I'm going to talk to you today about epigenetics, genetics, and behavior. And just want to acknowledge the various funding agencies that have funded our work over the years. And we're going to start by talking about individual differences in behavior and why we're all different. And I'm going to move in and talk about some of the Drosophila fruit fly work. I promise you it won't be boring. And then we'll move on to a broad view of epigenetics and how people have thought about it with respect to behavior. And then we'll go into what we think about what epigenetics might be doing now in response to behavior and what some of the open questions are. So I'll go back to, hopefully there's time, some of the fruit fly research that we've done that's causal, linking the importance of behavior, of epigenetics to behavior and how it might work. So that's the plan for today. So as Harold said, there's been long arguments about this nature-nurture dichotomy, which is really a dead argument now. And originally, it was thought that everything was determined, that it was nature, which we now think of genes influencing differences in behavior. And zoologists, biologists really had that point of view, as you can imagine. And then psychologists had the nurture view, that it's our experience that influences our individual differences in behavior, starting with the idea that a child is born with a blank slate and the environment writes on it. And then as time went on, people said, well, it's a little bit of genetics, it's a little bit of environment, and you add them together in an additive kind of way. But that didn't explain differences very well either. And so now we think about what's called gene by environment interaction, where genes give us a predisposition to behave in a certain way, but that's dependent on the environment, on the condition of the conditions in the environment. And to such an extent that the genetic predisposition can be reversed or changed by the environmental experience. And so it's not fixed in that sense, but we like to think about it as a probability of behaving in a certain way, depending on the environment. I'm using that word environment, of course, so loosely, because what the environment is, how it changes, what's important, what experience gets embedded in our biology is going to change according to our age, what we experience, etc. But biologists like to think of environment. Okay, so that's the history of it. And when I came into this, I was an undergrad student here in third year, and I really liked genetics because it made a lot of sense. And I was fascinated by animal behavior from watching animals from the time I was 3 or 4 years old in the creek collecting tadpoles on to just watching animals and people. And I wanted to merge those two things in a third year project that I was doing in a course in developmental genetics. And the prof at the time said, you cannot do genetic analysis of behavior because behavior is not a trait. It's not a phenotype. It's something that can't be measured. And that was the common thinking at the time. It was too complicated to do that kind of work. And so determined as I was, I said, well, I really want to do this project, and this is development. How about if I look at the fruit fly maggot, the larva, which is developing, and so it will be a course in developmental genetics, but my focus will be behavior. And she said, do whatever you want. And so I went to Brunswick Avenue, where there were some pear trees that weren't sprayed. I collected rotten apples, and I carried them down the street in bags back to the lab. I picked out the fruit fly larvae, found out that they were Drosophila melanogaster, the species that really a lot of genetics was known about. And I tried to figure what I should do with these animals. And because I had worked in a mouse lab in the summer, helping out, I made a tiny little open field for a larva, and I decided I'm going to look at how much they move and how much they eat when they're on food and when they're not on food. Just before I get to that, individuals of the same species display remarkable variation in behavior, even in identical conditions, but the molecular mechanisms that underlie this variation are still poorly understood. And in that fourth year project, I had a Petri dish. It was very low cost science, and it had a yeast paste on it. And I had another Petri dish that was agar. The yeast was food, nutritive, and the agar was not. And I took different larvae. I took larvae from that fruit of the same age, and found that there was a bimodal distribution in how these animals moved on this dish in food. And some of them moved a lot, and some of them moved significantly less when there was the feeding environment there, but in the absence of food, they move the same. So again, a difference in behavior conditional on the environment. And I got my old Rogers thesaurus out, and I thought, what should I call these guys? And I ended up calling these rovers and these sitters, and submitted the paper, got a B+ in it. And that was the beginning of the 40 years of research that myself and my students have done. And also we know that when there's patches of food present, just looking for my little cursor. Oh, it doesn't matter. When there's patches of food, rovers move from patch to patch, sitters go to the nearest patch and stay there. And so over time, we ask this question, is there a genetic contribution to these differences in path length, the amount that these guys move on food, the rover-sitter difference? And we did lots of genetic analysis to show that there were a lot of genes involved, but there was a major gene effect that was big enough that we could map it and track it. And so we followed that major gene effect. And eventually, my first PhD student identified this gene called foraging that was responsible to a large way to the rover-sitter differences. And there were two alleles of that gene, one we called rover and the other we called sitter. And this gene is known as, it's a signaling molecule, a cGMP-dependent protein kinase, and almost all organisms have it. We have it. And as time went on, we found out that it's a modulator of behavior. It doesn't only affect movement, it affects other things. It's pleiotropic. That's the genetic term for it. It has multiple functions. So it affects what I told you about food-related movement, but also how much they eat, how fat they get, their response to pain, learning and memory, stress, sleep, social behavior, and that this gene's role in behavior carries over from the time it's a little larva into adulthood. And so we've been studying this and trying to understand how it's regulated. How does this gene do so much of this? How does this one gene do so much? And I'm going to talk to you about that. The gene makes an enzyme that's found in the brain and the body. I said it's called PKG. That's what we're going to use as a short form. And interestingly, this gene is responsive to the environment. And what I mean by that, and I'm going to talk to you about that, is that a gene is a piece of DNA. So DNA is this long, long sequence. And one of our chromosomes is made up of a really, really long sequence of DNA that's smushed together like my curly hair. And then it's a chromosome. And on that long DNA sequence, there's a piece that is a gene. And so we have like beads on a string, different genes on that DNA sequence. And that DNA is making RNA. And when we say the gene's responsive to the environment, under certain environmental conditions, this foraging gene makes more RNA, and on other environmental conditions, it makes less. And this is true for rover sitter. Although rover has more RNA in its brain when it's well fed, when you food deprive it, it gets a lower level in regions of its brain, just like sitter. So we have this genetic predisposition that one should be a rover and one should be a sitter. But when we chronically food deprive these animals, their expression, the amount of RNA made from that DNA sequence that's a rover or sitter, becomes the same. They're both low in that case. And the social environment changes gene expression. If you deprive them acutely, it's different than if you give them chronic deprivation. So you can begin to study not just how the environment's interacting with the gene, but at the molecular level, what's happening to the RNA, to the protein. Where's it happening? Is it in the gut that's important? Is it the fat? Is it certain neurons in the brain? Is it glia, something we're looking at now, another kind of cell? So when I say the gene is responsive to the environment, we're not thinking about a fixed deterministic piece of DNA. We're saying, depending on the environment, this gene changes the amount of the RNA transcript it makes. Okay, And the gene expression, how much of that RNA is made, is regulated by epigenetic processes. So that's how we're going to get into that discussion. And our work and others have shown that the genetics, the DNA sequence, interacts with the epigenetic modification. So if we go back to nature nurture, the genetics is the DNA sequence, and it's not, I'll show you, that the epigenetics is nurture, which is what most people have been thinking. Instead, epigenetics represents the gene by environment interaction. So where, I have to tell you what epigenetics is first, but where the epigenetics leaves a mark is dependent on the DNA sequence. And that's one of the things that we discovered, but I'm getting ahead of myself because this gets me very excited, all this stuff. Okay. So it isn't only that this gene plays a role in foraging and feeding related behaviors in fruit flies, but also in nematode worms, honeybees, ants, and there are now more than 100 papers and maybe 50 species that have studied this gene in various ways. And sometimes there's genetic differences like in the fly and the nematode, and other times there is not a DNA sequence difference. Instead, when a honeybee is young and it's a nurse, it has a lower level of expression of this gene. And as it gets older and it becomes a forager and goes out and forages for food, the expression of foraging gets higher. So sometimes this gene changes its level within the lifetime of the individual. And in other cases, we have genetic differences, but even in those genetic differences, there's responsivity to the environment. So what about humans? So this is our first endeavor into looking at this, but to understand this, you need to know what a single nucleotide polymorphism is, which we call a SNP. And so here's two people, and this is a DNA sequence for whatever gene. And mostly the DNA sequences look pretty similar between these people, but in some spots, there's a different nucleotide. So here, there are 4 nucleotides for DNA sequence, G, C, A, and T. And in this particular case, this individual has an A in this spot, and this one has a G in this spot. And we can track these differences by doing the sequence of the DNA and seeing, does this one have an A or a G, and what about how it behaves, for instance. So some psychologists at Waterloo, James Dankert and Abby Scholler, James is a neuropsychologist and Abby is a social psychologist. A couple of their postdocs heard me give a talk years ago and said, we're sure we have rovers and sitters in humans. And I thought, oh, okay. And they told me that in social psychology, I think it's social psychology, people study the regulation of behavior, and they categorize some individuals as locomotors. They're always on the go. They move on. They don't sit still. I'm probably one of those. My husband's in the audience and he accuses me of never being able to sit still, and their brain is always on the go. Whereas, so that's a locomotor, but the assessor is the individual who sits around, weighs the pros and cons before they act, and eventually makes a move, right? Like if we're going to buy a TV, I say, buy this TV. Alan says, okay, I'm going to research it. And soon we get the best TV, which wouldn't have been the TV that I would have got because I needed to get it over with, right? And move on to the next thing. And so many of you probably experienced this with your partners or your friends. Anyways, so this self-regulation, being a locomotor or an assessor, seemed like an interesting phenotype to look at in humans. And we found a polymorphism in the human foraging gene. This is the number of it. That just says something about the location of it. And we saw that it also affected gene expression in a region of the brain, important for many things, but also regulation. And we gave the students who were part of this study a form to fill out, which had a scale of 12 questions, which they could answer. How far along are they on the locomotor scale from low to high? And how far are they on the assessor scale from low to high? And then we did the genetics, the snipping, to see, did they have AA, CA, or CC? This is again, just those single nucleotide polymorphisms, and they were on this scale, more locomotor this way and more assessor. And we found significant differences in their behavior. And then we went on to look at this. So in fruit flies, adult fruit flies, I'm going to tell you this later, more about it, and the epigenetic regulation of this. You can put a fly in a dish and put 4 drops of food and film them for 5 or 10 or 20 minutes. But if you film them, let's say for 10 minutes, you find the sitters hug the edges. They mostly hug the edges. And every now and then they venture into the middle and grab a drop of food. Whereas the rovers are more, I'm going to talk very loosely and anthropomorphically, maybe risk takers. And you can imagine a spider or a praying mantis lurking around the edges. And so the rover takes more of a risk and it goes more into the middle. And so with the same period of time in this dish, the same degree of food deprivation, one moves a lot into the center and one hugs the edges. And so when we look at this boundary bias, how much they hug the edges, in humans, we found a difference. And the way we measured that was the students got to sit in front of a computer screen and they developed a foraging assay where these little cherries, I guess, came onto the screen and the student had to touch it, right? And the question was, what strategy did they use? Did they go around the boundary and then jump in to get the cherry? Did they, in a systematic way, look and then get the cherry? Or did they have a strategy like the rover where they went in the middle and it was more mixed? And what we found is that also this boundary bias seemed to be significantly correlated, this is all correlation, with their genetics. And so we're moving on to study this more. It doesn't mean other genes aren't involved, environment, many things. It's just, at the moment, it's an interesting story. We don't know anything about mechanism, but it's a fun, interesting story, I think. Okay, so let's go and look at this fruit fly gene, because we need to know more about this before we move into epigenetics. So I said earlier, how is it that one gene can do so many things? How can it accomplish so many functions? And we know that this is really the rule, right? Because when you think about, for instance, drugs you might take, there's side effects, right? Well, what are those side effects? Those side effects, a lot of them happen because that drug that you're taking, that it might influence a gene, are influencing other things as well. So this foraging gene acts in muscle in the heart, right? And so if I wanted to take a drug, it also affects learning. So if I want to take a drug that might affect some aspect of learning, I would have to be careful because this gene also plays a function in the heart and the gut. And so it helps to understand how that gene regulates all these different traits. So for the foraging gene, we figured it out. And this was done by Aaron Allen, who was a former PhD student in the lab, who's a postdoc at Oxford. And he did a lot of the molecular work. So this is the DNA sequence, like I told you. It's very big for a fruit fly gene, 40 kb. And he found that this gene is modular. There's 4 different, what we call promoters, 4 different places where transcription can happen, where RNA can be made. And this promoter makes these RNA transcripts, and this one makes these RNA transcripts, and this one makes this, and this one makes all of these. And those transcripts get put in different places at different times and have different functions. And so even though it's one gene, and this is common for human genes as well, there's modularity of function, and you can study it in flies by breaking it down in this way. You can get rid of one of them and see what happens to the phenotypes you're studying. You can get rid of the whole gene, which he did, and see what happens. You can put more of these transcripts in, but not the others. You can put less of these transcripts in the fly in a certain area at a certain time. So you can do these studies that take this kind of behavior genetics research beyond association and correlation into causation. And so one of the quick things it wasn't quick, I'm going to tell you it quick, but one of the things he did is he used this fancy technique to delete the entire 40 kb sequence and made a larva that was missing in the gene. And it lived up to the point of being a pupa, the little cocoon. And then he asked, well, what happened to these different larval behaviors that we study? And what he did is he then put the gene back in different doses. So the first one, it was missing. And then he put in more and more and found that as he increased the dose of the whole gene, he got more longer path length, like Rover. And sorry, this mouse is super sensitive. And as he increased the dose here, they ate more. And as he increased the dose here, they became thinner. And that's the same function that this gene has in the mouse in terms of fat storage and breakdown, and also in humans. And so here we have 3 different phenotypes, and already we're seeing that the dose of the gene, what happens depends on the phenotype. And just to give you one more little glimpse of this before I move on to epigenetics. So what else Aaron did is he said, well, we have those subset of transcripts for each of those promoters. Where are those transcripts going? And for the first one, it went to neurons in the larva. And the second, he found the transcripts in glia. So in the middle of the larval ventral cord, there are these glial cells. And in the third, it was in another kind of glia, a glia that wraps the whole nervous system called perineuroglia. And in the last, it was more in the optic lobe. And then when he manipulated it so that certain transcripts went only to these certain regions, he found that he was able to modify the Rover-Sitter larval path length for promoter 1, the you know what, I need my glasses, but anyways, fat for promoter 3 and food intake for promoter 4. So not only was the gene modular, and there were subsets of RNA associated with each of these modules, but where those transcripts got put also had a modular function, and we could associate the behaviors we were interested in with the promoter and the transcript and the place in the larval life that it was important. So that's just showing you how we've been studying this behavioral pleiotropy. And if I was giving you a straight heavy duty fruit fly talk, which I'm not, I would show you that we've mapped many of these different phenotypes to these different promoters, pain, nociception, larval path length, fat, food intake. We're working on sleep, all of the different ones I told you. And what's really interesting is that they're all modified by different epigenetic regulators that I'm going to tell you about. And so in the case of the moving in the arena, hugging the edges or going in the middle, it's promoter 4 transcripts, and it's regulated by this epigenetic modifier, and nociception as well, but not Rover-Sitter path length. So we're figuring this out and how it's regulated. Okay, so now we're going to take a breath and go into epigenetics more, but I want to give you a little bit of a history on why this group that I used to co-direct, called the Child and Brain Development Group at the Canadian Institute for Advanced Research. I co-directed it with Tom Boyce, a pediatrician researcher from UCSF. So when we first became part of that group just under 20 years ago, there was a Canadian epidemiologist named Clyde Hertzman, who was very concerned with the socio-economic gradient in health. And at that time, when epidemiologists were doing work, they thought about SES as something that you can not study, but adjust the data according to SES. But SES was something interesting in itself in terms of human health and well-being, but people didn't think about it so much in those times. Now it's so obvious when I tell my first-year students, I teach a class for non-science students. It's really fun. In first year, when I tell them this, they go, what? But Clyde put this up when we were all together, and he said, I don't understand this. There's a socioeconomic gradient in health. And so it doesn't almost matter whatever you measure. If this is SES from low, so that would be the poverty end, up to very high, the extremely wealthy, there's this negative correlation between almost any trait you want to measure. There's some exceptions, single gene traits like cystic fibrosis, Huntington's disease, but there are very few examples of that. So 99%. And so if it's a chronic condition, asthma, ear disease, injury, physical inactivity, even literacy, we see this gradient. And Clyde said, somehow experience is getting embedded in biology. It's going under the skin. How is that happening? And so that's when this group started to think about, well, maybe there's a mechanism and maybe it's epigenetic. And just before I get to that, and you know this, all of you, there's health risks, and we have some MDs in the audience. There's health risks associated with early adversity and low SES. And this doesn't mean that every family at low SES has abuse, strife, emotional neglect, and harsh discipline. But on average, there's more of this. There's many, many stressors in a family living in terrible poverty. And as a result, we end up with health risks, depression, drug abuse, anxiety, diabetes, heart disease, obesity. And we have individual differences that people have studied in neuronal and endocrine responses to stress, brain development, and even later life functioning to do with the immune system. Now people are interested in studying guts, microbiomes, the little guys that live in the gut. And of course, we are interested in prevention. Now, what's also very interesting is that it's not that all people are affected the same way, right? And so, you know, the just so stories, there's twins in a family and they have a rough life, and one does extremely well and one doesn't, right? And those just so stories with the sample size of one don't mean anything to scientists and scholars in the audience, except it's a just so story. But when you actually look at this in a statistical way, there are individual differences, and some individuals thrive almost no matter what. It doesn't mean they don't carry problems with them, but they appear to thrive. And Tom Boyce has called these the dandelion children. These dandelion children can grow in the cracks of sidewalks and they're still gonna flower. If they come from a really poor environment, I'm using that term loosely, or they come from a highly enriched environment, they do pretty well. And you might think, if you're a dandelion kid, or do you have a dandelion kid? And so that individual is not the same no matter what. It depends on the environmental context. And so the dandelion kid would have a straight line, and the orchid kid would do better than the dandelion kid at very great, under great conditions. And so there are these individual differences. And generally speaking, scientists talk about averages all the time, right? And averages are not so helpful to us. Whether we're going to use an average and then treat females the same as the average we got from males, or an average of white men that we then use to make decisions about Black women, for instance. And so we have to be careful about that. And sometimes the individuals that lie at either end of the distribution are the most interesting of all to tell us an interesting story and maybe give us a hint on what we should study. So I just wanted to tell you about them. There's a book called The Orchid Dandelion Child. That's a popular book that Tom wrote recently. And it's fun to read and interesting. So brain development, just so you can think about this, I know some of you are experts in this, like Daphne. The brain is, and Harold's a neurophysiologist. Brain development is an engineering marvel. There's in humans 86 billion neurons, 10 to the 14th connections. Your brain sculpted by a lifetime of experience, especially in the first few years of life and during adolescence. Early life and adolescence are thought to be critical periods in development where environmental input needs to come in order for that sculpting to occur. And then from a genetic and molecular point of view, experience changes the expression of our genes. I've said this already. Okay, so now we're going to get into epigenetics, the first word in the title of my talk, but I wanted to set the scene for you. So if you look here, this is the DNA sequence that I showed you. And epigenetic means it acts above the DNA, epigenetic. And what happens with many of these mechanisms is a little chemical tag, like a methyl tag, is placed on the DNA. It's not part of the DNA, it's placed on it. And it can be in response to environment that it's placed on it, for instance. And in the case of DNA methylation, it's placed right on the DNA sequence. And in the case of histone modifications, which we study, it's placed on something called the histone tail. So this DNA sequence, it gets wrapped around these histones, nucleosomes. That's what these little balls are. And when it's in the chromosome, it gets all compressed and wrapped around. Think of one of my hairs or a string, and you keep twisting it and twisting it and twisting and twisting. You know what happens? It gets really small. So that's what's happening here. And when it's all twisted up, you can't access any part of it easily. So any molecule like a transcription factor that's telling the gene, okay, make more RNA, it's very hard for that molecule to get in there and bind the DNA and do it because it's in the configuration of what they call closed chromatin. But when it's opened up, then you can get access to the DNA and then make RNA. Now, with these methyl tags, it makes the DNA sequence less accessible. It makes it all tied up, and the molecules can't get in there to make the DNA express RNA. Okay, so this is what epigenetics is. There's different kinds of it. We're going to talk about DNA methylation, histone modification. There's also these tiny little microRNAs. They're little pieces of RNA that are in the cell, and once the RNA is made, they bind to it and take it out of the system. And we might I hope get into talking about transgenerational inheritance and how epigenetic might act for things to go from one generation to the next. And these little RNAs are what people think could be involved in the sperm of the male to transfer information like trauma from genocide or the Holocaust, but this is just correlational at the moment. Okay, epigenetics isn't new. Developmental and cell biologists have been studying it for decades, and they were studying it from the point of view of cell differentiation. So every cell in our body has the same DNA sequence, right? But some of it becomes a brain cell, some becomes a liver. How does that happen? And so epigenetics is involved in that cell differentiation. But this behavioral epigenetics is pretty new. And so I'm going to tell you a little bit about the origins of that to help develop the ideas. So DNA, you can think of it as like books packed in boxes and stacked in a library. It has limitless potential to inform and inspire, but it has to be unpacked and read. And so the reading of it is the gene expression or the RNA, making the RNA. That's one way to think of it. I don't know, I guess I'm losing track of what I'm showing. Okay, so there the little girl is reading it. She's opening it, she's reading it. And when we have an epigenetic mark, like that DNA methylation I showed you, it keeps the chromatin, it keeps that chromosome very tight, the DNA tightly bound and all twirled, and it's hard to read. So you have an epigenetic mark, it keeps it all tight, and the chromatin isn't open and you can't get other molecules in there and make RNA. When it is not, when the epigenetic marks are not there, the DNA is in an open conformation, and it can be, the regulators can get in there and it can make RNA. And so the early work showed that early adversity makes some genes difficult to read because of these, they thought, these epigenetic marks. And the main gene that was studied was the glucocorticoid receptor gene, which is involved in how we cope with stress. Any of you who might know about the hypothalamic pituitary axis, it's involved in that, and cortisol, it's involved with that. If you don't know about it, don't worry about it. we want to stay calm, right? So it affects how we cope with stress, how our brain develops and works, and how we fight disease. So over the many years, people have associated epigenetic variation, that is, in this case, DNA methylation, finding DNA methylation marks on certain genes with stress, drug use, social interactions, environmental toxins, smoking, hormones, and nutrition. And these are all correlational. Now, the first study that really got people interested in this, it was, and had to do with behavioral epigenomics, is from Michael Meany and Moshe Zip at McGill. And what Michael noticed is that these are rat mums. He didn't notice they're rat mums, they are rat mums, but he noticed that their style of parenting, their maternal behavior differs. Some rat mums lick and groom their pups a lot, and others lick and groom their pups much less. And if you look at all the rat mums he had, you found a normal distribution in licking and grooming. So you have the whole range. But for Michael's studies and his student studies, they only took the extreme differences. So they chose a bunch of rat mums that they knew after hundreds of hours of watching these guys were high licking and grooming mums, and ones that were low licking and grooming mums. And they asked the question, well, how is it? What happens to the babies that were licked and groomed a lot, like the Jewish mother maybe? And I don't know if our children are licked and groomed a lot. And then those that are licked and groomed less. Okay, so this experiment they did, they got a mom with her babies. They were all licked and groomed a lot. The blue shows these are babies that come from a mother that licks and grooms a lot. And here we have the other end. Sorry, I got this. This is low licking grooming is blue, high licking grooming is yellow. And same thing. And then they asked the question, well, what if this blue guy that is genetically similar to this low licking and grooming mother gets swapped in a cross fostering study with these two, so that you have a yellow mom who licks a lot, looking after a litter that has two babies that from the original low licking and grooming. So they were sort of trying to get at something that might be genetic and something that might be environmental. And they asked the question, does it matter who the actual biological mom was, or does it matter how you were treated? That's the experiment they did. They're psychologists, so they didn't control the genetics, but this was their experiment. And what they found is that it mattered how you were treated. And this is an example of the gene expression, and they had the biological mom is first in this histogram, the foster mom second. And you can see that what the biological mom was didn't matter for the gene expression from this glucocorticoid receptor. Instead, it's what the foster mom was. So here we have high foster mom, high licking and grooming foster mom. They had higher gene expression from this receptor. And here we have the low licking and grooming foster mom, and they had low. And so this suggested that it was how you were treated that affected gene expression. And they went on to look at DNA methylation at a certain site in that gene, and said that this is epigenetic, and it's a mechanism for nurture in this case. And there's a bit of human work that again is correlational, but shows the same kind of pattern. This was done on postmortem brain banks that Gustavo Turecki has at McGill, where you had individuals who had committed suicide and that did not come from a history of abuse, those that committed suicide and did come from a history of abuse, and then controls. And here we have the amount of methylation that you could see on this same gene and now in humans. And we can see that the methylation was high for the individual who committed suicide with abuse, and the gene expression was low. And this is what the relationship you would expect, because the DNA methylation, the result of that is less gene expression, as I explained, because of the the availability of the DNA sequence. And then this went on to people doing more correlational work and looking at the DNA sequence in some of these genes and sometimes in the whole genome for this one epigenetic mark, the DNA methylation mark that I told you about, the little tag added onto the DNA. And Rachel Yehuda led a lot of this work and showed that the individuals who were in the Rwandan genocide, them and their children had different DNA methylation, different amounts of DNA methylation, more in the case of people with genocide. Famine, the Dutch winter famine was studied, and then went on to look at child abuse and neglect and the Holocaust. And my father was a Holocaust survivor, my husband's parents too. And so in some of these studies, they looked at the children of Holocaust survivors, whether they were male or female. And interesting results that show similar patterns and again, correlational. And so the people who criticize this area, criticize it because it's associations and correlation, and people have been urged to try and make this more causal. And there's a paper that we wrote, I don't know if I gave it to Harold, but I will, that talks about how one might be able to do that using animal models and studies that follow individuals through time and things like that. But it's clearly a challenge to do this. Now, one other thing I want to mention is that when you think about all of this, you have to think about timescales when we're thinking about correlation or causation or what genes and environment do, when they interact. And there's different timescales in this kind of work. So there's a neurobiological timescale, and this is milliseconds to seconds. This would be a neurophysiological timescale. And in terms of critical period, that's the kind of timescales that we're concerned with. And then there's minutes, minutes to hours, where you can see in the brain that when someone has an experience, the gene expression, we can measure that, changes. And you can see there's different colored dots according to what genes are expressed. And then at a daytime time scale, we have circadian rhythms. You all know about that, sleep, wake cycle, etc. And then for critical developmental periods, you see that brain plasticity changes depending on the age of the developing child. And so even before birth, the child is responsive to the environment, to auditory signals, work from Janet Worker. And there's this window of opportunity where the child needs this environmental input. Then there's language, and there's many different critical periods of language, and then also higher cognitive function. And so when we're thinking of critical periods in development, and the role that epigenetics plays, which we know it's involved in the opening and closing of critical periods, it's months to years. Now, in terms of what I was just referring to, we also have intergenerational and transgenerational. Now, intergenerational would be an example of the mother is stressed. There's a fetus inside the mother, and that fetus is affected by that stress. And so that child may be born with different characteristics. So for instance, there's some data that a child that is carried and born into a war zone may have different behaviors, may shift their attention often, their risk-taking might be different, etc. And people could tell a story that this has to do with what happened in utero. And then, and then there's transgenerational inheritance. And this is from one generation to the other. Here we have a Holocaust survivor talking to their grandson. And so we can say, yes, individuals are affected by their parents' experience or their grandparents' experience. We know that. We know a household, for instance, where the parents are survivors of genocide would be a different environment than another household. The parents didn't go through that, maybe walking around, now I'm telling just so stories, but with a feeling of dread that stays with you. But we don't know that epigenetics is involved in these things. And it's very challenging to figure this out and to separate transgenerational experiment from intergenerational experiment. And one of the reasons it's challenging is because, let's say I'm this baby inside this mother. Well, my eggs are already made when I'm the fetus, the female fetus inside the mother. And so my eggs, which will give rise to my daughter, in this case, my mother's granddaughter, would have been exposed to my mother's experience. And so if you ask about epigenetic inheritance and with women, you have to consider that eggs are there to begin with, whereas for men, the sperm are constantly being replenished. And so to make the argument that there's an inheritance of behavior from one generation to the other, and it's epigenetic, down the female line, you need to go more generations than down the male line. And then the other issue around this is that the zygote, once you have egg and sperm coming together, all the epigenetic marks, almost all of them get wiped out. This is a normal process. There might be a few that hang around, we don't know. And so if that's the case, how is this patterning from history getting transmitted? And at first, people thought it's DNA methylation. We know those marks are pretty much wiped out. Then histone modifications, we know they're wiped out. And now people are studying these tiny, small RNAs that are found in sperm and trying to track them to see if that continues into the next generation. So if you hear people talking about inheritance through the ages and epigenetics, inheritance of trauma and epigenetics as a mechanism, we actually don't really know that. We know in plants that there are many examples of that, but in humans, it's possible there's some, and there's some reviews on it, but it may not be that common. But that's not to say that there isn't a kind of inheritance of behavior, the way that you're treated, that goes from generation to generation. That there's a lot of very nice social science work and psych work. But here the question I'm bringing up is epigenetics the mechanism for that? And it could just as easily be the behavior of parents, the way you're treated, the behavior of your community that has you growing up in a certain, your nutrition, your neglect, abuse, etc. So something to keep in mind. I just want to go back and tell the fly story. How am I doing for time? Okay, I'm taking my time because I'm trying to make this clear. I hope it's clear. Okay, I don't know if I can do this in 5 minutes, but I'll do my best. So, okay. So this is a paper that we published recently that talks about the biological embedding of experience and provides a primer on epigenetics and really goes into the idea that we should be careful how we are talking about this. And for human work, we need causal information. And it gives you the summary of epigenetic mechanisms as we know them now. And I've only talked about DNA methylation. That's the one that's most studied because it's the easiest to study. So here we have the DNA sequence, as I told you, and we have this methylation mark that likes to go near C's and G's, and it's a little tag, and it changes the ability for genes to be expressed. And you have another kind of epigenetic mark. There's a lot of different ones. And then those are called DNA modification. Then there's chromatin modifications, and that's a little tag that goes on this histone tail where the DNA gets wrapped around it. And I told you about that too. It makes it more or less accessible. But there's also two others, and the one is about these tiny little RNAs that sop up RNA that's being made, and long coding RNAs, they're being studied now. And this one is hardly studied at all. It turns out that the RNA also gets modified. One of my former students, Rna Interference, who just got hired in biology at UTSC, she's taking on this topic. And it's very tricky because it's hard to measure it, but this may be very important too. Okay, so let me just show you how we can address this causally in fruit flies. And there's the foraging gene that I told you about, and there's this histone modifying gene that puts a little methyl tag. It's called G9A. It regulates gene expression, is what people thought. And when we go back to this foraging gene, I already told you there's 4 promoters, and this is work from Rna Interference. a former PhD student, and we found that this G9A is methylating, adding little methyl tags to those histone tails in the area of all 4 of the promoters. And we didn't know if any of them are going to be important, but we want to ask the question, does epigenetic modification account for these rover sitter differences in behavior? And we have that same little assay I told you about. You can see it more clearly here. The fly comes up the middle of the dish. There's a trap door, pokes its head out, comes out, and it can hug the edges, or it can come in and get drops of food. In this case, there's 4 drops of food. They're dyed blue, and we can see who's there and who ate the food. And these are the trails I showed you before. And there's a cost of coming into the open. You might be grabbed up. Sometimes we have jumping spiders near our assays to see. The undergrads really are entertained by this. So I'm not. Yeah, I am too. Okay. So what about the behavior? So we see, as I told you in this assay, that the rovers will eat more drops of food than the sitters. And that doesn't matter how long you do it for. And because this is genetics, we're able to completely take away this G9A gene, the one that does this histone modification. Genetically, we're able to delete it. And so we have a rover sitter difference, and we can ask, if we completely get rid of G9A, do we still see this difference in behavior? And we don't. And so that tells us there's something important about G9A for the rover sitter difference in behavior. And it's not that there's more or less G9A in rovers and sitters, because we measured that, and the gene expression is the same in rovers and sitters. So that doesn't explain it. And so that's the behavioral data I show you. And then we asked, well, what about gene expression and methylation? And we found just here that for methylation, how many of these tags were put on, that it was the 4th promoter where we had the same type of pattern. The rover was methylated more than the sitter. And when we made the deletion that took away this gene, this epigenetic gene, the difference went away. And the same for gene expression. Whoops, where's my gene expression? Ina cut up the fly. So he looked at these patterns in the brain and the gut and the ovaries and the carcass, and found that for the brain and ovaries, there was less gene expression RNA in the brain of rovers and sitters. When we took the epigenetic modifier away, it didn't differ, and the same was for the ovaries. So that helped us narrow down what tissue could be involved. And then finally, to get at this causally, we have something called RNA interference. And so we can take the transcripts only from that promoter 4, and we can reduce them or get rid of only those set of transcripts. And when we do that, when we reduce the RNA transcripts from promoter 4 using RNA interference in sitters, you get a sitter fly that behaves like a rover. So getting rid of only those transcripts from promoter 4, we could change sitter into rover, both in terms of how much it ventures into the middle and also how many drops it gets. Now it eats. And then I know I'm going through this quickly, but I'm trying to give you the idea that there's causal particular manipulations we can do to ask, does this epigenetic modifier affect gene expression? It does. Where does it affect it? What tissue is important? So here is the last little bit on this is there a SNP? Is there a single nucleotide polymorphism that is somewhere near this promoter that is somehow interacting with this epigenetic modifier? And we think there is. We think, oh my God, this is quite the fancy computer. Okay, here we have a sequence of A in rover and C in sitter, and that is a place that a transcription factor binds to change gene expression. So this told us that the DNA sequence interacts likely with the epigenetic modifier, and that was one of the first publications that showed that. So this is just finishing up now. When we started, we talked about nature and nurture, and that then we went to genetics and epigenetics, and it seemed that epigenetics might be a mechanism for nurture, but now we know that the epigenetics actually responds to the DNA sequence. And at a statistical level, this shows you data where we have, this is mums that had babies, and many of these DNA methylation regions were studied, and prenatal experience was looked at, maternal smoking, depression, BMI, infant birth rate, BMI of the baby, and birth order, but also the genetics, the SNPs, the DNA sequence was looked at. And then statistically, they said, what of these factors, genetics, environment, gene-environment interactions, influence the DNA methylation? And it was found that it's the genetics is very important for where those DNA methylation marks are going to go, and the gene by environment interaction, but not the environment alone. And so this tells us that that earlier idea that epigenetics is an explanation for nurture alone, which is still the nature-nurture dichotomy, is not the case. And there's more and more data showing that. So in summary, individuals of the same species display remarkable variation in behavior, even in identical contexts. The molecular mechanisms that underlie this variation are starting to be unraveled. And I hope that I was able to discuss with you about individual differences, how, what, when, and why, from the perspective of epigenetics, genes, and behavior. Thank you for listening.